Hello, and thank you for attending today's webinar, Monkey Business Meets Heart Science, a New Era in Cardiac Safety Screening with Non-Human Primate iPSC-derived Cardiomyocytes, presented by Ncardia and Fierce Biotech. I'm Andrea, and I'll be your host for today. Before we begin, just a few quick housekeeping items. To learn more about our speakers, please visit the speaker bio window. If you accidentally close a window, you can reopen it by clicking its name in the top navigation bar. Additional resources can be found in the handouts window. This webinar is being recorded and will be available on demand within 24 hours. And now I have the pleasure of introducing our speakers. Joining us today is Ilse Veldkamp-Baak, Scientist Manufacturing Technology at Ncardia, and Silke Schwangberg, Associate Director, Cardiac Discovery Services at Ncardia. All right, we're ready to start. Silke, over to you.
Thank you very much, Andrea. And I'm happy to take you through the key takeaways of our webinar today. So you will learn about NHP in vivo models as a cornerstone of translational research. We will introduce iPSC technology and how that can fill a critical gap in translational and in vitro studies, and actually how a step back might help to achieve this. Then we will introduce our NHP ventricular cardiomyocytes, which are highly pure, functional after freezing, and show stable beating activity. And then we will also show you some pharmacological results from different assays. And that should help you to understand that these cell models could be suitable for predictive safety and toxicology studies. So now let me introduce non-human primate models to you in translational research. So they always have played a critical role in research and non-clinical drug development for decades because of their genetic, anatomical, and physiological similarities to humans. You would assume that with the general increase in vitro technologies, this kind of in vivo study would be on the decline, but that is not true. Especially for NHPs, the numbers are actually rising. This is partially due to the rise in novel modalities like biotherapeutics, which might not work at all in other species. The human immune system, for example, is in many cases very different from that of our standard in vivo species, which often leads to NHP as the only option to test these therapeutics in animals. But despite the utility of the NHPs, they are not a flawless research model. Growing ethical concerns, rising costs, supply shortages, and biological differences are placing increased pressure on the industry to seek viable alternatives. As mentioned already, the use of NHP understandably raises significant ethical questions and public scrutiny, which in turn led to increase and more stringent oversight and restrictions in possible use of NHP. This makes it an expensive and time-consuming model. On the other hand, there are serious supply shortages. Breeding of NHP is complicated, and the demand cannot be fulfilled. As an example, in 2021, only 1/3 of the requests could be fulfilled worldwide, although I have to admit that the number was extraordinarily high due to the vaccine development. The biological differences that I have already mentioned contribute to challenges in translating NHP findings directly to humans. Any case of inconclusive findings between in vivo NHP and the first clinical data would usually require going one step back and run mechanistic in vitro studies. But there is a lack of in vitro models for NHPs. If you look at the ATCC catalog, NHP cell lines are really scarce. I could only find 26 of those. And primary tissues is not an option due to the ethical concerns. So we know that there is still a need to run in vivo NHP studies. But in April of this year, the FDA issued their roadmap to reduce animal testing in preclinical safety studies. This strategy paper recognized the limitations of the current models and outlined a strategic approach to increase the number of new approach methodologies, or NAMs. Now, what is a NAM? In fact, this can be many different things, not even necessarily all in vitro. Using telemetry instead of open chest electrodes in anesthetized animals is a NAM already. But more specifically, the FDA listed human cell-based in vitro assays, organ-on-chip systems, and in silico models as possible approaches. Overall, the goals of these initiatives are enhanced predictive accuracy of drug safety and efficacy, reduction in research and development costs, acceleration of the drug approval process, and minimizing the use of animals in research. If you want to build a good organ-specific in vitro model, you need to have cells available that are mimicking their natural counterparts in the body. Often, the usual cancer cell lines are not even close to that, so primary cells might be used instead. Unfortunately, while some models can be easily generated with primary cells, like liver cells, cardiomyocytes, and especially human cardiomyocytes, are very difficult to source and to work with. Since the invention of induced pluripotent stem cell technology, iPSC-derived cardiomyocytes have been one of the first cell types to be implemented into in vitro assays, including also more sophisticated NAMs. These assays are now routinely used for both efficacy and safety studies. In a recent paper from the FDA, a total of 99 unique studies using iPSC cardiomyocytes have been reported between 2012 and 2023, with a steady increase observed since 2017. Most of these were safety studies. So obviously, human iPSC cardiomyocytes are out and working. So why do we think that we need NHP cardiomyocytes? The problem is, again, the translation between a human in vitro model and large animal in vivo data. And NHP are just one of these animals. It is already challenging to find concordance between species, but here you also have to account for the in vitro to in vivo differences. Over the next minutes, we will introduce our NHP cynomolgus iPSC-derived ventricular cardiomyocytes to you. These cells can become the missing link to bridge the gap between human and animal cardiac in vitro safety studies. They eliminate the ethical dilemma, so there is no need to sacrifice the monkey to get the cardiomyocytes. They can be generated in unlimited quantities and can be easily integrated in your standard in vitro test battery. Plus, they offer the additional possibility of gene editing and disease modeling. This makes the cells ideal for cross-species mechanistic studies that could validate findings between preclinical in vitro and in vivo studies, de-risking the drug development by providing predictive in vitro data before moving into the in vivo studies, and of course reducing the animal use in alignment with regulatory expectations. I will now hand over to my colleague Ilse, who will show you how the NHP iPSC cardiomyocytes have been produced and qualified.
In order to manufacture the ENSITE non-human primate cynomolgus ventricular cardiomyocytes, we actually first had to source non-human primate cynomolgus iPSCs. We have sourced 2 non-human primate cynomolgus iPSC lines from the German Primate Center. They originated from healthy male cynomolgus monkey and were reprogrammed using episomal vectors, so there's no integration in the DNA. If we're looking at the image on the right, you can see that the morphology of the non-human primate iPSCs is very similar to the human iPSCs, showing clear nucleoli and a high nucleus to cytoplasm ratio. These cells were thawed, expanded, and we've done quality control on both of the lines. This quality control included a cross-contamination test, confirming the species of the. We've also checked for pluripotency. So here we see an example for one of the 2 non-human primate iPSC lines, showing that SOX2 and OCT3/4 expression, which are both pluripotency markers, is very high, so over 95%. Moreover, we confirmed that both lines were negative for mycoplasma, and we have done some pathogen testing, and the pathogen test included a very large panel of non-human primate-specific pathogens. So when both lines passed the criteria of the quality control, we expanded them further, and we made sure that we cryopreserved the iPSC lines in a bank that we could use to start with when we're starting every differentiation experiment. Our first differentiation from non-human primate iPSCs towards ventricular cardiomyocytes was performed in amber bioreactors. Amber bioreactors are controlled bioreactors, which means that you control the pH, the temperature, the stirring, and the dissolved oxygen. In the system, you can fit 24 small vessels that contain 14 milliliters of medium. Using the system, we tested 12 conditions in Duplo to optimize the differentiation protocol. So each protocol that we tested actually consisted of the same basis. So first we have done a pre-differentiation in which we inoculated the iPSCs into the bioreactors and gave them some time to form iPSC aggregates. This is followed by pre-cardiac differentiation that consists of two different phases. When we're treating with two different media. And then at the end, there's cardiomyocyte differentiation until we process the samples. Like I mentioned, we have tested 12 different conditions. So we included both the NHP-C iPSC lines. We have tested different growth factor combinations, but also concentrations. And we also played around with the timing of the compound treatments and the differentiation end day. On the differentiation end day, we processed the cells, which means that we dissociated the aggregates into single cells, and we have performed flow cytometry and determined the cardiac troponin T expression for each of the conditions. We then selected the optimal conditions, so the ones showing the highest percentage of cardiac troponin T, and brought them into our intermediate scale manufacturing bioreactors, which are the DASBox bioreactors. Also, the DASBox bioreactors are controlled bioreactors. In the DASBox bioreactors, we repeated the same procedures as were performed in amber, and we cryopreserved the batches. With that, we actually produced a test batch. There was one batch showing a very high percentage of cardiac troponin T, a very good recovery and viability after cryopreservation, and therefore we were really happy that we had generated this batch. Looking at the purity of the cells, we noticed that the cryopreservation did not affect the purity, and we observed beating monolayers in the postal analysis. We also did the first test for functionality, and we observed electrophysiological safety in a multi-electrode array assay. So with this, we produced the test batch in intermediate scale, but we actually wanted to go into an even larger scale to produce a catalog batch. So therefore, we moved to our BioFlow bioreactors, which is a large manufacturing scale. The bioreactor can contain 1 liter of medium, and we use this to produce the catalog batch. So during the production of the catalog batch, we do a lot of in-process control. And the in-process control has also been done in the previous step, so in the optimization of the differentiation, to make sure that we have a very standardized and robust process, and also to make sure that we can reproduce what we're doing. So this in-process control includes imaging and aggregate sizing. Also, the cell counts and viability over time are very important. For example, we also look at the intermediate marker expression. So if we look at the brachyury T, which is a marker of the mesoderm stage, we expected the expression to go up around day 2 and then go down again. This is what we see in the 3 bioreactors that we show here. And something else that we keep track of is metabolites. So for example, LDH, lactate, and glucose concentrations. We keep track of that over time. And with that, for example, we can measure the glucose consumption of the cells. So like I mentioned, we have generated now a catalog batch using this bioreactor. And when we were cryopreserving this batch, we had to dissociate the aggregates to single cells. And after that, we used controlled rate freezing to cryopreserve the cells. And this actually means that you gradually cool your cryophiles in a programmable rate. And with that, you actually prevent damage from ice crystal formation. And thereby you preserve viability and you ensure consistency of your cells. From 1 liter BioFlow, we generated a batch of over 500 million assay-ready non-human primates cynomolgus ventricular cardiomyocytes. Now it's very important to also perform quality control of the NHP-C cynomolgus ventricular cardiomyocytes. Therefore, we set it up a timeline for the postal analysis. So what we do in the postal analysis is we thaw the cells at day 0. We count the cells and we determine the viability, and then we seed the cells. We culture them for 2 days, doing a refresh on day 1, and then on day 2, we harvest the cells. We can do cell counts, a viability measurement, and then FACS, or we can perform immunocytochemistry. So one of the very important criteria of the batches is the number of viable cells or assay-ready cells on day 2 and the viability of the cells. So when looking at our catalog batch, we observed at thawing 4.7 million recovered viable cells per vial with a viability of 82%. After 2 days of culture, we harvested 1.2 million assay-ready cells per vial at a viability of 99%, showing that there's a very strong recovery and viability after thawing the non-human primate ventricular cardiomyocytes. Moreover, we observed visible beating clusters within 72 hours of seeding. Besides the cell numbers, it's also very important to look at the expression of the markers. So when we're looking at the cardiac troponin T expression before cryopreservation, at thawing, and 2 days after thawing, we can see that the cardiac troponin T expression remains really high, over 95%, showing that the cryopreservation and the culturing of the cells does not affect the purity of the cells. The expression of the cardiac troponin T was also confirmed using immunocytochemistry. So 2 days after thawing, we performed an ICC and stained for alpha-actinin and cardiac troponin T. And we can see a very nice staining over here. So besides the expression of the markers, it's also very important that the cells are functional. So therefore, we decided to include MEA electrophysiology in our quality control. This is a very physiologically relevant in vitro high-throughput platform, and with the multi-electrode array, you can study the behavior of cardiac cells in disease modeling, but also drug efficacy and safety assessment. So how does it work when we're seeding the cells? So we have a cell suspension. We seed the cells drop-seeded on MEA-96 well plates with embedded electrodes. So we can see an example here on the right showing that each 96 well contains 8 electrodes, and those electrodes can capture the electrical activity. And after that, we can analyze some parameters, for example, the beat rate, the amplitude, and the field potential duration. Some other important parameters for reliable MEA studies are the number of active electrodes. Of course, we prefer to have all the electrodes in the well active. Something else is the intra-well variability, which is the variation in electrical signals in the same well over different time points. And also the interwell variability, which is the variation in electrical signals from different wells on the same MEA plate. So actually, these important parameters we have used to set up some criteria for our baseline MEA measurements. So this was actually the first time that we were going to do multi-electrode array using these cells. So we still had to optimize the timelines and the seeding densities for this assay. There's actually 2 ways to use these cells. So we can either do 2 days of pre-culture, which means to thaw the cells, seed them in T25 flask, refresh them on day 1, and then on day 2 you harvest the cells, seed them in the MEA plates, refresh every other day until day 6, on which you can do the baseline measurement. But there's also the option to use the cells as plate-ready cells. And this means that right after thawing, you seed your cells in the MEA plates. Then you refresh every other day and do your baseline measurement on day 6. So both procedures we have performed using our catalog batch, but also our test batch. And we actually have some criteria for our MEA analysis. So for example, for the beat rate, we would like to see it between 20 and 80 beats per minute. The interwell beat rate variability, we would like to see below 25%. The intra-well beat rate variability below 5%. And we are aiming for 8 active electrodes per well. So if we look at the catalog batch, used in the plate-ready timeline, you can see that we tested 3 different densities. So there's 5, 10, and 15K cells per 96 well. If we're looking at the 5K cells per 96 well, you can see that the average active electrodes is 7, which is actually too low because we're really aiming for 8. When we're looking at the 10K, we can see that it passes all the criteria that we set. And when we're looking at the 15K cells per 96 well, we can see that the intra-well beat rate variability gets really high, around 30. So therefore, we determined that for the plate-ready timeline, it's best to use 10K cells per 96 well in your assay. Then when we're looking at the pre-culture timelines, so the top one, you can see that for the catalog batch, we have tried 3K and 5K cells per 96 well. And for the test batch, we have tried, so this one is the one from the DAS box. You can see that we tried 5K and 10K cells per 96 well. If we're looking at the 3K and the 5K densities, we can see that it's passing all the criteria. If we're looking at the 10K cells per 96 well, we can see that here as well, the intra-well beat rate variability gets too high. Since the 5K cells per 96 well passed the criteria for both batches, we decided to move forward with that density. So we've used those optimal densities to check the baseline recordings. And here you see a comparison between the pre-cultured cells and the plate-ready cells. And you can see that there's complete electrode coverage. The settings of these images are the electrical activity is over 300 microvolts. So the cells are really active. If we're looking at the peak shapes, we can see that there's a very consistent peak shape for both the ways of culturing the cells. And if we look at the beating patterns, we can see that the beat periods remain very consistent over time. Then we wanted to see whether we could see the expected pharmacological responses. So we have treated the non-human primate cynomolgus ventricular cardiomyocytes with the compounds that we also use for the human cardiomyocyte quality control. So we have treated the cells with nifedipine, dofetilide, and isoproterenol. If we're looking at nifedipine, we can see that the cells stop firing at all concentrations for both the pre-cultured cells and the plate-ready cells. When we're looking at dofetilide, we can see extra spikes within the traces, also for both formats at 10 nanomolar dofetilide, and these extra spikes show actually that there is arrhythmia in there. When we're looking at isoproterenol, we can see a dose-dependent increase for both the pre-cultured and the plate-ready cells. And when we're looking at the standard deviation, we can see that the error bars are smaller for the pre-cultured cells compared to the plate-ready cells. And this actually means that the data is slightly more robust when the NHP ventricular cardiomyocytes are pre-cultured. So we decided to move forward with the cells that were pre-cultured and do some comparisons in drug responses of the NSAID NHP cynomolgus and human ventricular cardiomyocytes, which Silke will tell you more about.
I will show you now the results from some assays where we directly compared drug responses in the NHP cardiomyocytes and our standard human ventricular cardiomyocytes. We were convinced that the NHP ventricular cardiomyocytes were active in the MEA assay and respond to the usual suspects. We wanted to test them side by side with our human cardiomyocytes in two different assays. Assay 1 is again the MEA that Ilse introduced to you already, and where we can measure the electrical activity of the cardiomyocytes. But we also wanted to know if the contractility of the NHP cardiomyocytes can be quantified. And to this end, we used an impedance assay. This is not a direct force measurement, but impedance technology can be used for label-free real-time and kinetic measurements of cellular function under full environmental control. And this technology is sensitive enough to detect the subtle morphological changes due to contraction and relaxation of cardiomyocytes. We ran both assays with compounds that are known to act acutely on the cardiomyocyte, which are mostly ion channel modulators, but also compounds that act chronically. And that could be by causing structural damage or inhibiting certain proteins. Due to the differences in the two cell types, the protocols look slightly different. For the two protocols, you can see in the MEA assay that we have a shorter culture, pre-treatment culture time, for the NHP than for the human cells. And that is also true for the impedance assay. Since we run the impedance assay both for acute and chronic compounds, we actually seeded the cells on two different plates. And then for the acute measurement, we only measured within the first 24 hours, whereas for the chronic measurement, we also measured after the following days of the treatment. When comparing the MEA data for the two batches of NHP cardiomyocytes and then two batches of human cardiomyocytes, we observed some interesting differences. In isoproterenol treatment, which is a selective beta-adrenergic agonist, and which you can see on the left graph here in the upper panel of the slide, we could see the dose-dependent increase in the beat rate in both species. But what you can also very easily see in this graph is that the magnitude of the effect was very different, with the NHP showing less increase than the human cells. For nifedipine, which is a calcium channel blocker, this was reverse. The NHPs were more sensitive and showed firing arrest at all tested concentration, whereas the human cells first showed a decrease in the activity before they also finally stopped beating at the higher concentrations. And then for dofetilide, a hERG blocker, the effect was more nuanced, depending on the concentration, but matches in vivo observations that the NHP seemed to react at lower doses than the human cells. What we could observe, though, is these arrhythmia forming in both cell types, which were very pronounced at the 10 nanomolar concentration. To measure the differences in contractility, we used the impedance assay. We first were interested to see if these are differences between the two cell types under basic culture condition. Interestingly, and matching what we learned in the MEA assay, we had to seed less NHP cardiomyocytes to obtain a similar signal quality than our human cardiomyocytes. We also learned that the NHP cardiomyocytes require higher frequency of media changes, even with the lower seeding density, indicating to differences in metabolic activities. As you can see from the traces here on the right side, there are some differences in the shape of the contraction signal between the two species, and that also translates into the calculated numbers. You can see easily that the beat rate and also the contraction amplitude are quite similar between the two species. However, despite the similar beat rate, the time that it takes for each cycle of contraction is lower in the NHP cells compared with the human cells. And that is what you can also see here in the traces, where if you look at the distance between these two peaks, this looks different in the traces. What we also observed is that we indeed can also do chronic compound testing in the NHP cardiomyocytes, where we have a total assay window of 10 to 12 days. We first analyzed acute acting compounds and tested both dofetilide and nifedipine, also in the impedance assay. As you can see in the graphs here at the top of the slide, with dofetilide, we observed a dose-dependent decrease in the beat rate and an increase in the IBD50, so the intra-beat duration at 50% of the repolarization height, which indicates that one cycle of contraction takes longer under dofetilide than without. The very high standard deviation here at the very end of the assay with the higher concentration is due to the arrhythmic effect that we could observe in both cell types. And this is also shown here in these little waveform traces. What you can also see here is that we observed arrhythmias already at very low concentrations in the human cells, whereas we only observe clear arrhythmias at a higher concentration in the NHP cardiomyocyte. Interestingly, the shape of the contraction signal that we could see with the impedance assay did change already at earlier concentrations without showing signs of arrhythmia. Nifedipine in the impedance assay induced beat arrest at similar concentration. However, a reduction in amplitude was seen in the human cardiomyocyte at a lower concentration than in the NHP cardiomyocytes. We also tested two other compounds, which is moxifloxacin, which is also a hERG channel blocker. And with the moxifloxacin, the NHP cardiomyocytes were more sensitive than their human counterparts. Last but not least, the sodium channel blocker quinidine induced different effects in the two cell types. We observed an increased beat duration and arrhythmia in the human cardiomyocytes, whereas the NHP cardiomyocyte showed an increase in beat duration only at the lowest tested concentration, but actually a decrease and an impressive fibrillation pattern at higher concentrations. Next, we also analyzed chronic cardiotoxicants, where we showed the effects after 24 hours as well as after 3 and 5 days of treatments. In general, the NHP cardiomyocytes in this assay were more prone to arrhythmia observed with some of the compounds, which were usually not necessarily shown also with their human counterparts. The tyrosine kinase inhibitor sunitinib, where you can see the data here in the upper panel at the top of the slide, showed concentration-dependent cytotoxicity after 5 days of treatment in the humans, but not in the NHP cardiomyocytes. We could observe an increase in the IBD in both species, but if you look here at the magnitude of the effect, you can see that this was much more pronounced in human cardiomyocytes. So here we can clearly see that at all days that we analyzed, we can see an increase in the IBD already with a relatively low concentration and a huge increase here with the highest concentration. What we observed in the NHP cardiomyocytes is arrhythmia after 5 days of dosing, and this was not visible in the human cardiomyocytes. The anthracycline doxorubicin induced concentration-dependent cytotoxicity in human cardiomyocytes after 2 days of treatment, but only starting at 3 days of treatment in the NHP cardiomyocytes. Both species showed a beat arrest after 2 days of treatment at similar concentrations. We also used 2 very differently acting drugs. One of those is blebistatin. Blebistatin is a myosin ATPase inhibitor, which effectively uncouples the electrical and mechanical action in the cardiomyocytes. So you will still see firing, but they should finally stop beating. The NHP cardiomyocytes only stop beating after 5 days of treatment, where we observed changes in the human cardiomyocytes very early on. Finally, we also tested the hERG trafficking inhibitor pentamidine, which induced cytotoxicity at the highest concentration in both species, but at day 4 in the NHP versus day 3 in the human cardiomyocytes. The functional effect was different, though, with the NHP cardiomyocytes being more sensitive and showing arrhythmia and beat arrest, where the human cardiomyocytes only showed an increase in the IBD, so in the intrabeat duration, or the time that it needs to go through one contraction cycle, at the tested concentrations, but no real arrhythmia. So even with this very small set of compounds, we could already observe species-specific differences, mostly in the sensitivity to a drug, but also in the way how the cardiomyocytes reacted. To summarize all of this, the ENSIGHT NHP cardiomyocytes are the first ever NHP cardiomyocytes successfully developed and validated. They are highly pure, viable, and they are stable after cryopreservation. Both the MEA and impedance assay demonstrated robust and stable performance of both the human and the NHP cardiomyocytes. And both species showed expected pharmacological responses to acute or chronic cardiotoxic compounds. However, we also observed species-specific differences like arrhythmia signatures or the chronic sensitivity, which are indicating that the NHP cardiomyocytes are a highly valuable translational model, especially for safety screenings. So in essence, the ENSIGHT NHP cardiomyocytes open a new era in translational cardiac safety testing, providing unprecedented insight into species-specific drug response. And with that, I will hand back to Andrea to finalize the session.
Yes, that's all the time we have for today. I'd like to thank our speakers for their thoughtful contributions and thank our audience for joining us. If you submitted a question during the presentation, we will do our best to follow up after today's webinar. As a reminder, a recording of this session will be available on demand within 24 hours. Thank you for joining us, and we look forward to seeing you next time.